Hello, everyone. Good morning, good afternoon, good evening, depending on where you are from in the world. Our speaker is from the US, Washington, DC to be particular, and it's morning that side, it's 8 a.m. And it's 3 p.m. Afternoon in South Africa. So I understand that we come from different sort of time zones. So thank you so much to all of you for joining us for the last webinar for 2021. And the title of the webinar, as you can see on the screen, is Population Descriptors in Genomics Research, what classifications are used? And this seminar will be provided by Dr. Shawneequa Callier, who's an associate professor in the Department of Clinical Research at the George Washington University School of Medicine and Health Sciences, and training in law, bioethics, and healthy in genomics. I'm gonna sort of give a few ground rules just to make sure that we don't disturb our speaker and that we have a well-run webinar. I'm going to kindly ask that you remain muted if you're not asking a question. And if you do have a question, kindly raise your hand or type it in the chat box if it's a burning question. And I'll obviously field the questions to our speaker today. Just to also say, this webinar is being recorded. It's also played live on Facebook. So if you have an issue with any of those, please do advise, or you can leave the room, because I understand that consent is quite important. And that's actually one of the things that is going to come up today. I met Dr. Callier a few weeks ago at the IHCC workshop, which I co-organized with my training and workshop committee that I co-lead there. And she was giving a presentation and she was part of a panel. And it was really engaging and interesting and informative. And myself and my PI, Professor Nicola Mulder, were like, we need to invite Dr. Callier to give a talk to our HTA Bionet and African community. And when I emailed her and I got in touch, she was really excited. And I'm really grateful for agreeing to give this talk. So just a bit of sort of background on the abstract and Dr. Callier's biography. And I'm not going to waste time. I'm just going to quickly sort of mention a few lines. And if you want to read more about her, please Google her, go on the website, and go on the ad that I sent out. So the abstract is quite short, and I'm going to read the whole thing. It's a growing number of diverse participants engaged in genomics research. It is crucial to advance standards for properly labeling populations. However, the processes for classifying the populations that researchers include in biomedical research are often unclear, inconsistent, and poorly justified. This presentation today will examine the use of population categories in research, discuss ongoing research in this area, and describe challenges with the current population labeling system. So that's what you can look forward to in this presentation. She has an illustrious resume or CV. So I'm not gonna obviously cover the whole CV because it's gonna take the whole webinar for me to do that. So I'm just gonna mention a few things. Dr. Shawneequa Callier is an associate professor in the Department of Clinical Research and Leadership at the George Washington University School of Medicine and Health Sciences, training in law, bioethics, and genomics. Professor Callier teaches courses in bioethics and serves as a special volunteer at the Center for Research on Genomics and Global Health, National Human Genome Research Institute, National Institute of Health, or NIH in the US. On several occasions in the past, Professor Callier taught genetics and the law as an adjunct professor at the George Washington Law Faculty. Prior to joining the George Washington University faculty, Professor Callier completed a postdoctoral fellowship at the Center for Genetic Research Ethics and Law, an interdisciplinary center for excellence by the National Human Genome Research Institute, and located in the Bioethics Department of Case Western Reserve University's School of Medicine. I could go on introducing her, but I don't want to waste much more time. Thank you so much for agreeing to give this talk. The floor is yours.
Thank you so much. Can you hear me okay?
Yes, Prof, I can hear you perfectly.
Wonderful. Thank you so much for that kind introduction and for the opportunity to join you and your colleagues at H3 Africa BioNet today. My background to start, although I'll be talking about genomics policy, my background is in bioethics and law. I have the pleasure of collaborating with geneticists, including my colleagues at the Center for Research on Genomics and Global Health. However, of course, the opinions expressed are my own and should not be construed as the views of GW or NIH or any of my affiliate institutions. And I declare no financial conflicts of interest. So I was asked to build on my presentation to the IHCC a few weeks ago and to discuss race, ethnicity, ancestry, and data collected to explain what labels are used in genomic studies of populations. And so I'll discuss goals to harmonize cohorts in global genomics research. I'll describe an IHCC cohort study and discuss the challenges of using population descriptors in research, as well as opportunities for the global genomics research community. To start, I'd like to share that I became interested in this topic because of my work on law, race, and pharmacogenetics. The United States Office of Management and Budget census categories are used in biomedical research in the United States, and often these same United States census categories are exported internationally to describe global populations, especially when companies want FDA approval. In the United States, the categories we use have changed over time based on political and social factors. In 1790, the categories were white, free, or slave. And that's how we described populations then. In 2020, the census included new areas for people who identified as white or Black to fill in more details about the ancestries or groups that they identified. For example, a person who checks white might write in Egyptian or Lebanese. So NIH requires researchers to certify that they have made sufficient efforts to enroll diverse populations in research and that the resulting cohorts are adequately diverse using these cohorts. FDA recommends a standardized approach for collecting and reporting race and ethnicity data. Published qualitative research shows that investigators believe that using racial categories in their studies will help them get funded and published, and that researchers feel incentivized to use racial OMB categories when analyzing results, even if they don't have to. More research in Africa, one of the reasons I'm really excited about being here with you today is because more research in Africa by African scientists provides opportunities to influence the development of global vocabularies and to develop terms that better capture what populations and ancestry scientists are studying, and also to improve our understanding of global human genomic diversity. So let's turn to some background. We are witnessing right now unprecedented interest globally in diversity, equity, and inclusion in genomics research. In response to alarm bells our colleagues have sounded and continue to sound about the lack of inclusion of diverse populations in genomics research, and in particular, a certain type of study that we all know about, genome-wide association studies. And so this interactive dashboard, which I checked yesterday, called the GWAS Diversity Monitor, tracks the diversity of participants across all published genome-wide association studies. While the sizes of studies have expanded significantly, samples representing populations with European ancestry still dominate GWAS. Demographic and geographic representation are limited. So in these images, the GWAS Diversity Monitor uses a mixture of limited and very broad continental, regional, and racial and ethnic categories. They allow for a simple snapshot of continental and regional underrepresentation and historical exclusion. You can also see that the terms European, African, and Asian represent supercategories that cover large segments of the globe. These categories are too broad to depict underlying population diversity, but they show that there is sparse and non-existent representation of populations around the world in published GWAS. This image shows that the availability of GWAS in diverse populations is growing. For studies of groups that are of European ancestries, data have now been compiled for several 100,000 individuals. The emphasis on populations with European ancestries over time, however, has caused a cycle where it is easier to publish data about European ancestries. So this brings me back to the landscape today. Many in the global genomics community recognize the need to increase diversity, equity, and inclusion in genomics research, participation, collaboration, and leadership. Importantly, efforts to build capacity to conduct genomics research in Africa are taking place alongside an international effort to also harmonize and standardize how scientists categorize populations. So the funders of genomics research want data to be accessible across borders, interoperable, and reusable through standardized vocabularies and reporting practices. Less clear is how to achieve this goal as more diverse populations are included in data sets. What population descriptors should we use? In its most recent strategic plan, NHGRI stated its aspiration to move beyond social population descriptors such as race. This is a clear goal outlined by NHGRI that is welcome by those who would rather focus on scientific categories like genetic ancestry. This bold prediction raises the question, what happens next when we move beyond race? Many scholars are concerned that emphasis on continental categories will still racialize genomics. For instance, for researchers and publications in a way stuck looking at ancestry in terms of continental clusters, there may be a tendency to compare or racialize these categories. European ancestries, for instance, would be equated with white and African ancestries with Black. Of course, our world is not black and white, and people with European and African heritage live in both regions. So thinking through a bioethics lens and a legal lens, how will these categories promote inclusion, fairness, equity in healthcare when applied to clinical practice in the future? How will they help us understand science and move away from eugenics, which we know has no scientific basis? Since sharp boundaries do not exist along continents, ethnic and linguistic groups, or racial and ethnic groups, the task ahead of standardization seems challenging. In Africa, given the immense diversity in ancestry, geography, politics, and culture on the continent, scientists working on the African continent are well positioned, of course, to inform these discussions about the limitations of broad categories. And I just really look forward to the solutions that come out of the work that you're doing as a part of H3 Africa. So one potential area to address is the lack of definitions communicated in publications. Empirical research shows investigators almost never define how data were selected to fit within racial, ethnic, or ancestry categories in publications. When genetic researchers analyze genetic ancestry, it is often unclear in publications how ancestry was constructed or defined. I have here a rather old study published in 2011 and a more recent one published in 2020. I'll give you a chance to look at the quotes. So Ali Khan and her colleagues reported in their systematic review of the literature that authors often fail to define race, ethnicity, or ancestry. To assess how researchers' reporting changed over time, they compared their findings with a prior study highlighted in blue, which analyzed genetic research articles between 2001 and 2004. Now highlighted in yellow, they found increases in the percentage of articles explaining why samples and participants were grouped by ethnicity or ancestry or race, which is good. And also an increase in articles justifying why these particular populations were studied. And I think that's really important. There's so many different factors and categories that can lead to differences in health outcomes and that can be relevant to populational differences. And so if we are going to use certain population descriptors, we should explain in our publications why we chose those descriptors, as well as explain how those labels came about, why we chose the labels that we chose. And the authors concluded that consistent and comprehensive reporting on human populations is imperative. They found there was no change over time, and you can see this highlighted in red in terms of articles defining the terms race, ethnicity, and ancestry in the context of their study. In the 2020 paper, the authors explain that when geneticists, and they also talk about consumer genomics, when consumer genomics companies talk about ancestry, they are really talking about genetic similarity between populations and individuals. So this gets at what do we mean when we use the term ancestry in our publications? What are we communicating as to the public, to readers, and of course to people in different fields, people like me who have backgrounds and that are not necessarily genetic backgrounds. So they, my battery's running low, although my computer's plugged in. So hopefully I will not have a, hopefully my computer won't shut down. Let's keep going. Just check it 1 second.
Prof. Callier, I think it might be frozen. Yeah, sorry about that. I think her battery was low. So can we please, oh, she's coming back. Hi, Prof. Prof. Callier, you're on mute. Prof. Shawneequa, you're on mute. Are you aware? Oh, I can't hear you. You're speaking, but Prof. Callier, can you type? Because I can't.
Yes, I'm so sorry. I'm having trouble charging my computer. If this happens again, I'll just change computers.
Okay, sorry about that. Technology can fail us sometimes, but that's okay.
I know, I'm sorry about that. All right, so let me go back to where I was. From current slide. Can you see the, have I resumed now?
Yes, Prof. This is the slide you were on.
Okay, great. All right. So where was I? Let's see. All right, so I was at the point of explaining part of the figures. So the authors explained that an ancestral recombination graph, or ARG, is used to depict representation of genetic ancestry in a study. And that ARG construction is part of many methods for making inferences about demographic history. And so they note, and I quote, "In any real population, individuals will differ in their ancestry, and the true ARG will be extremely complex." And also another quote, ultimately, all descriptions of ancestry, whether approximate or based on reconstructing an ARG, imply some arbitrary choices. And so in this figure, they show how methods that summarize genetic variation among samples, such as a PCA, are frequently interpreted in terms of ancestry. Some of these methods allow individual genomes to be represented as combinations of reference populations or ancestry-like relationships. And I want to take a moment to just go over some of the points that they make, additional points, and relate that to how I see that as someone with a background in law and bioethics. So measures of similarity are always sensitive to the selection and labeling of reference populations. By inferring ancestry based on genetic similarity, we tend to emphasize and reify existing groups, hiding important connections between populations and individuals, possibly giving the false impression that these processes are not biased when they are. They say geneticists may find it useful to refer to clusters of individuals as real populations as a shorthand and as an aid to discussing the results. But people in other fields could become confused. The limited or biased sample size of reference panels also finally limits the usefulness of these genetic similarities. So again, my takeaway applying a law and bioethics lens is that there is an assumption that we all mean the same thing when we use these categories. And this has implications for fair representation. Sometimes such labels are important to study questions and results, but it would be important for investigators to explain what made these categories important in the particular study and how was ancestry determined. So now I would like to switch gears and talk about research collaboration with my colleagues at Duke University and the IHCC. We came together, we've emailed members of the IHCC cohorts because we believe that IHCC is poised to contribute to this discussion given its global reach, variety of cohorts, and research on genomics and environmental factors. And so a few of us came together over the past year and developed a survey with goals of better understanding how our colleagues are defining populations in their cohorts. And what we hope to do is also assess IHCC cohort opportunities and challenges, collecting, defining, and reporting data, and use findings to inform discussion that we're having about harmonization and standardization of global cohorts. So this is an IRB approved study, but I am not reporting formal results. We are still in the data collection phase, in the beginning phases of data collection. This is just a snapshot of what we are seeing so far for some, but not all survey questions. And if you're here today and you're a member of the IHCC cohort, please contribute and fill out a survey if you haven't already. Thank you to those who have. All right, so we administered this survey. So far, we've received responses representing 6 countries and 7 cohorts, 2 in the United States. Here are responses to the question, do you collect race, ethnicity, ancestry, or other data? Check all that apply. You can see the differences there. Then when we asked, please list the categories you use to collect race, ethnicity, ancestry data, please copy and paste the questions and categories from the survey. I'm sorry, I'm admitting people to the room. Please list the categories you use to collect race, ethnicity, and ancestry information. And so here are how people responded. We also asked about genetic assessment and how if they performed an analysis of genetic assessment or if they assigned ancestry to populations. Okay, so this gives you a sense of some of the variety we're seeing in some of the responses so far. Of course, we hope to receive many more responses and continue this work. So, we see some diversity in how cohorts define populations and use genetics to assess ancestry. But we can also see some challenges related to using population descriptors play out. So one challenge relates to how we use population descriptors. And it's that the categories we use can be very broad, like white versus Black. Also, the rationale or circumstances that lead to researchers using some categories are often not clear. This is especially true for populations that are less represented in research. So a quick look at this ontology lookup service is which is a repository for biomedical ontologies that is used for the NHGRI GWAS catalog, shows that categories are a mixture of broad racial, ethnic, regional, and continental groups. And there are a limited number of sub-Saharan African categories. A large number, as well as a great deal of specificity, is available for European populations. The ontology is an illustration, I think, of the work ahead for African scientists, for those of you on this call, in terms of representation and developing labels that we may find should be dynamic or related to time period. How do we capture changing populations? This is how we see and label groups now, but how will that change in 20 or 30 years as we collect more data and also as populations continue to intermingle as they have been doing for generations? Many populations are listed in the undefined ancestry population. Too many to list here. So looking toward the future, will our new population descriptors advance or hinder understanding about ancestry? Will they erase and marginalize some diverse ancestries or provide clarity on human genomic diversity globally? As the genomic research community, we should strive for equity in representation, definitions, and categorization. And we might have to acknowledge that these labels are imperfect and capture a wide range of heterogeneity. So before I go on, I'm often asked to define race. What does race mean? When it comes to race, people often ask for a definition, but there's no agreed upon definition for race. Race, ethnicity, and ancestry can be avowed, ascribed, or perceived. The twins pictured here may identify as biracial, but one may be treated as Black and another white when enrolled in a study. Looking towards the middle column, Dr. Fulwiley wrote that she is Black in America and white in parts of Africa. In the course of a plane ride, her race could change from Black to mixed if she flies from America to Senegal to France. In Chad, a person may identify socially with one ethnic group, but genetically have significant ancestry from different ethnic groups or reflecting Eurasian migrations. On Google Scholar, this article is cited by about 28 others, some of which talk about the need to conduct further studies on the diversity of ancestries in Africa, like this one. So how do we incentivize understanding of the complexity of identity and genetic ancestry? Answering that question will be important, of course, to the future of research. When thinking about ancestry, my colleagues often argue and say that genetic ancestry is a scientific term and that it should be free of racial overtones. The image published here in a paper by my colleagues at CRGGH uses different colors to represent 21 different ancestries that they identified. They identified a few new ones in addition to others that had already been identified, determined from 282 global population samples. And the figure simultaneously shows ancestry mixture within individuals, populations, and geographical regions. Ancestral heterogeneity, they argue, is common in the vast majority of individuals and samples collected, as well as within racial or ethnic groups. They emphasize that there is continuous genetic variation among populations and no sharp boundaries between groups. And here is a quote from the article: "Ancestry cross-classifies ethno-linguistic groups as well as continent and race." So I understand that one might ask, well, race can be tied to social factors. It's important to keep track of race and or certain ethnic identities. And I think those images that I showed very early on showing the underrepresentation in research, it illustrates how keeping track of certain categories can be useful to make sure that people of diverse backgrounds are included in research, assuming that we develop categories that truly reflect our underlying populations. But these categories also have implications for the groups within groups. And this is an article that's been out for quite a while, but it's an excellent example of study results that were originally reported according to really broad categories, where scientists found that individuals with European ancestry were likely to have an allele that caused an adverse reaction to the HIV drug abacavir. And actually in the United States, recommendations were made that people who have European ancestry or ancestries should be screened for this HLA allele. But then researchers looked at the prevalence of this allele in other groups. And so in this pharmacogenetic study of HLA alleles, there are medically relevant differences in the allele frequencies observed among the Luya, the Maasai, and Yoruba, who scientists are likely to label as African or even East or West African. There's diversity within the East African region. There are also significant differences between Chinese, Japanese, and Gujarati Indians, who scientists might describe as Asian ancestry populations. Thus, the broad labels we use have health implications because they can obscure biomedically relevant variation. And so I want to end and allow some time for discussion. What are some opportunities here? Opportunities to understand practices regarding population. Opportunities to assess ambiguities and inconsistencies in how cohorts collect, record, and report population data, and an opportunity to contribute to the discourse on global harmonization in ethical and scientifically appropriate ways. We need all hands on deck, global involvement in this effort. So that's all I have for now. Thank you so much.
Okay, thank you so much, Prof. Callier. That was brilliant, that was amazing, and that was informative. And I'm hoping that there's tons of questions, because I mean, I have a few myself. It's now time for question and answer session. So please, if you have a question, kindly raise your hand or type in the chat box, and then I'll read it to our speaker from today. So I'm going to give it a few seconds to see if there are any hands that are coming up. Comments, questions, concerns. Okay, so I'll start with my own questions. So you spoke about the collaboration between Duke University and IHCC, the survey that you sent out, and that you still need sort of responses. So I think I have tons of questions on that. Are you sending it just to IHCC members? Or can you share the link with us here? as researchers in Africa, but particularly also on the survey, did you define race, ethnicity, and ancestry for the participant? Yes or no? And do you think that has any or will have any impact on the result of responses by the participants of the survey? So that's the first question. And I'll stop there for now and see if there's anything from other people. Yeah.
Okay, thank you so much for the question. And thank you for those of you who have placed comments in the chat. One of my colleagues from IHCC is here, Dr. Melanie Cortez, who can, I hope, correct me if I say anything that's wrong. But from what I understand, we are starting by focusing on what's happening in the IHCC membership, which represents just so many cohorts across the globe, to get a sense of just what's going on in IHCC. And we are not defining race and ethnicity or ancestry for our respondents, because we want them to tell us what categories they're using and how they are categorizing populations. And again, the goal is really just so we can get a sense of what these diverse cohorts are actually doing.
Thank you so much for that, Prof. Callier. Are there any questions or comments? Oh, Melanie, go ahead. I see you.
Hi, Shawneequa. That was really, really good as usual. I had a question.
Thank you, Melanie.
I really liked your, the idea that you showed the ancestral ontology, and you said, the African descriptor are underrepresented versus the European ones. And then on your following slide, you were saying there's a need to be fluid when describing race and ethnicity, because, for example, the colleague you mentioned, depending on where she was, the label and descriptors that apply to her were changing. How do you think that's compatible or not? Or how do you think that could be handled when we consider, global data and global data integration? And I suspect if she's US-based, it could be that somebody in Europe uses our data set. And so how do you see that evolving? The fluid aspect is something that I find fascinating.
Well, I think you're pinpointing exactly a challenge we need to address. And that's the fact that researchers in different parts of the world use similar terms, but mean different things. So a researcher in France may use the term Black or mixed, or even in England, where members of, for example, some Asian groups might be called Black race. We will use the term race, but mean something different in France or in England than we do in the United States. And so if we have a global ontology and we have a term such as Black, how do we make sure that we really know what it means in a particular study? And I think what it shows is that researchers who are creating language and vocabularies should first define the populations, how they define them, and contribute to a discussion where we can make sure that we all know what each other means when we use terms, rather than assume that we know what each other means. So rather than assume that African ancestry on one part of the globe means the same thing or includes the same populations as African ancestry used by another scientist, rather than assume it, let's be clear about how we constructed the category, who was chosen to be in it, and why.
Okay, thank you so much for that. I'm just going to read. There's a comment from Professor Makani from Tanzania, who's saying, excellent talk, Prof. Callier. This is a terrific presentation. And there's a comment from Hala Abuzid saying, thank you so much for an interesting and informative talk. And there's a last one from Dr. Shakuntala by Chu, great presentation about a very relevant topic in research. Professor Rajram, please go ahead and ask the question.
Thanks very much. Can you hear me?
Yes.
Thanks.
Yes, Prof, we can hear you.
Shawneequa, fantastic presentation. Thank you very much. I think there's just a huge sensitivity with regards to using these kinds of, whatever you care as seen politically, especially where you're talking to a lot of researchers from the developing world. Again, I'm using a charged term. But it is my understanding that, you know, the terminologies we use, and especially as scientists, usually are context dependent. And if you're using a term, you've got to define why you're using that term. So if you're using the word race, you've got to define what it is. This is not, you know, race is a social construct as far as I'm aware. I'm a scientist, but that's my understanding. It's a social construct, and it probably started in, you know, during the colonial exploits of them and us. And those are those things, and these are these things kind of thing. You know, they're black, we're white, perhaps, and I'm oversimplifying. And the reality is the space we're in at the moment is where there is a genuine need with regards to showing that there are differences. I mean, as you showed in those beautiful vertical lines representing the different continental constructs of genotypes, you know, and the high level of heterogeneity, even in very small geographic regions. I mean, the ultimate is when we're referring to any, let's say, in this instance, we're talking about genetics and genomics. In this regard, you know, you're often talking about one locus, one single point in the genome, 3.5 billion base pairs, and you're referring to that, you know, heterogeneity at that level. And it is important for pharmacogenetics or whatever you're defining to say, well, that there are differences across the planet. And this is the reason why I'm using this terminology. So as long as we're able to use and understand, I think in my very simplistic understanding, you know, referring to differences for that particular reason, there has to be a reason why you're talking about something in terms of differences. And we've seen the high level of genetic variation across the genome. And these have huge impacts with regards to disease predisposition, susceptibility, and obviously response to drugs. So it is important to understand these. And I suppose as geneticists or as a scientist or as medics in the environment, it's really important for us to also understand this, but also to go beyond the sensitivity and really call it for what it is and use a common language. And try and construct that common language, which I'm presuming this is the start of. Thank you.
I completely agree. And so beginning this research looking at pharmacogenomics in a country where our racial categories are political and hierarchical and were created so that we could discriminate against groups and then later changed so that we could track discrimination. I'm really excited about building cohorts from other populations around the globe where the approaches to describing populations might be different, and also better understanding those approaches. And as you noted, if you are going to use a category, it is going to be context specific. So we should explain that. In addition, why use a racial or ethnic or ancestry category at all? Is percentage of ancestry linked with the outcome that we're trying to find or understand? In some cases, perhaps other factors, educational attainment, access to water, a person's environment may be more influential on a trait or the trait that we're studying. And so I think it's also important as we are collecting genetic and environmental data to also realize that if we're focusing on one particular category, we may be ignoring another particular category.
Okay, thank you so much for that answer, Prof. There's a question from Prof. Makani saying, what would be the simplest ontology in South Africa to define race, ancestry, or identity?
Well, I would turn that question to you. And, you know, when I provided the images in the very first, in the first couple of slides, it really showed dramatically how much exclusion there is in terms of GWAS studies anyway. And the fact that there are so many ancestries that we are not paying attention to or not studying. And so with the recent initiatives to build capacity for genomics research especially in Africa, where there is just so much diversity politically, environmentally, culturally, as well as in terms of ancestry. It's just exciting to think that there could be a real opportunity for those of you who are a part of these capacity building projects and others who are listening in, who may not be receiving the funding to do this work at the moment, but can comment on it, can engage. My colleagues here, you on this call, should be playing a leading role in telling us in other parts of the world, or contributing to these conversations about what these groups should be.
Okay, thank you so much for that answer, Prof. We still have time. We still have about 11 minutes. So if there are any more questions or comments, please go ahead and type in the chat box, raise your hand, and I'll give you the right to speak. Are there any hands or comments? Because I do have a second question, which I hope is my last one, but I wrote it down while you were presenting, because I found it's sort of like what's bugging my mind. So there's a school of thought, or some groups of scientists, that believe that science is apolitical and ahistorical. So they deny the fact that science is always in conversation with power. I'm thinking about things like eugenics, where categories such as race were used to harm the world. And I think obviously science has to grapple with that, that very painful history. So how do we challenge the notion that science is apolitical and ahistorical? Because I mean, some people just think it's all about observation, it's all about facts. They don't necessarily think that those scientists that are doing the research put their subjectivity in their studies as well, and their positionality as well. Thank you for that, for your answer.
Thank you for that question. Well, if we consider the way we group populations in the first place and the kinds of questions that we ask, those questions are politically and socially informed. And so I would simply respond that science does not exist in a vacuum, and that what happens in society impacts what happens in the lab. And so as we make diversity and inclusion and equity priorities in genomics research, I'm really enthusiastic about the focus on scientific leaders and bringing diverse people to the table to lead the scientific studies and ask the questions. NIH performed a study and found that scientists who asked questions about cell biology were more likely to be funded in the United States. And scientists who asked questions about health disparities and other environmental factors that lead to health disparities were less likely to be funded. Scientists that were Black or African American in the United States were more likely to have questions related to health disparities, and they were less likely to be funded. So there are social and political factors that influence the questions we even get to ask with our federal tax dollars in the United States and fund. And by having such underrepresentation around the globe, we are prioritizing diseases and questions that are important on the agenda in American and European and other nations with a high level of resources. And so the scientific results and the relevancy to the globe are impacted by those social and political factors. And I'll just say one more thing. We are living in a world where there's a great amount of mistrust. We see this with underrepresented, marginalized populations in the United States and indigenous communities. And by ignoring the political and social factors that have led to that distrust, and by not engaging diverse communities in order to assess how science and researcher can become more trustworthy, we are seeing firsthand populations not wanting to be included or participate in genomics research. And this has implications for science. It's not only about ethical fairness and access and justice, but also our goal as a larger scientific community to better understand global human genomic diversity. And if people don't trust the science if we don't begin to fix those relationships today, where will we be in 30 years, in 40 years? How will the science get done? And will people trust it if we continue to pretend that science happens in a vacuum?
Wow, thank you so much for that answer, Prof. Callier. Are there any more questions or comments before I wrap up the webinar for today? Going once, going twice, going 3 times. Oh,
can I just say one more thing?
Go ahead.
I just want to say thank you again. And I'm just really excited about looking forward to this particular group leveraging, utilizing, engaging with people and resources on the highly diverse continent of Africa, because I just think that there's just so much possibility in terms of what capacity building in African countries can do in terms of leadership in this area.
Thank you so much, Prof. There's one last, 2 more questions, I think. There's one from Hala in the comments saying, why do you think are reasons behind those variations in defining populations between the 6 research groups that joined your research so far? I think referring to the survey. Are they geographical, political, or religious? So that's a question from Hala.
We don't have the answer to that question. If I had to guess, I would say that these are categories that we're used to using, that scientists already use. We already use super categories a great deal and break people up into continental groups quite a lot around the globe. And it's a common way to describe populations. That has never been okay, even as we lack representation. But because there are concerted efforts now to diversify research and in Africa in particular, to include representation from diverse regions and locations in Africa, we need better categories that reflect the research results that we're finding and reflect your engagement with diverse African communities. So while I'm not sure it could be more than one answer to that question, it seems that we've been doing more of the same in terms of the labels that we've been using. And so now that diversity and inclusion is increasing, what comes with it is a duty to revisit, reimagine these categories.
Thank you so much for that. There's a comment from Prof. Makani saying, Sickle in Africa will be in touch with you to learn from you and from this work as well. So I think I expect a lot of emails. So I'll forward you the recording. Oh, there's a question from Nikki. Nikki, go ahead. Nikki?
Hi, sorry, yeah, I'm in the car. So we have in Shawneequa, we've developed an ethnolinguistic ontology for Africa, where it's
Nikki, I'm sorry,
to depth of all the existing ethnolinguistic. Yeah, you can't hear.
I lost you. You okay. Are breaking up.
So we've developed an ethnolinguistic ontology for African ethnolinguistic groups in Shawneequa. Is that a better way to represent the data, because it's based on actual linguistic groups that are sort of recognized rather than color of skin or something else?
Great question. So if one of the studies I referenced, the one that had the images of 21 different ancestries in 282 global samples, they used ethno-linguistic groups, and they used genetic ancestry to better understand and discover ancestries that existed in their samples. And they used ethno-linguistic categories to help clarify what they were seeing in terms of genetic ancestry. So according to those researchers, there are ways you can use ethno-linguistic groups and genetic ancestry to inform one another to better understand global genomic diversity. I know that you have an H3 Africa BioNet phenotype group. I know that IHCC, H3 Africa, and others are developing vocabularies and ontologies. And now is really ripe and a good time to assess how well these vocabularies and categories are working with populations.
Thank you so much for that. I'll share the email address, recording, and everything with you tomorrow, because we're gonna place the recording on YouTube. But for now, that I wanna email today, the email is scallier@gw.edu, scallier@gw.edu, and you can follow her on Twitter @slcallier. And yeah, I just want to say thank you so much, Dr. Callier, for a brilliant presentation. I think we have so much to think about and also debate about. Oh, thank you so much. So that's the Twitter account. Oh, thank you so much for sharing those. I'm really grateful, and I think the audience is also grateful. So thank you so much. For everybody.
Thank you.
This is the last webinar for the year. We're not going to have a webinar in December. We'll be hosting another one in January. If you have a suggestion of a topic that you want to hear, please email me. Or if you want to be a speaker, kindly email me and we'll speak on the side. And I'm going to be sort of setting out a timetable for 2022. So thank you so much for joining all our webinars for 2021. I think it's been a brilliant year in terms of webinars. Prof. Callier, we'll be in touch. Thank you so much. I'm really grateful. Goodbye, everybody.
Thank you all. It was wonderful to be with you today. Enjoy the upcoming holidays.
Happy Thanksgiving to you, by the way.
Thank you.
Thank you so much. Goodbye, everybody.
Bye.